As we age, many of us experience an unwelcome change, an increase in abdominal fat. While this transformation is often dismissed as a natural part of getting older, its consequences extend far beyond aesthetic concerns. The accumulation of belly fat is linked to a range of serious health issues, including a slower metabolism, diabetes, cardiovascular disease, and even certain cancers. Understanding the underlying mechanisms behind this weight gain is crucial to developing effective strategies to combat age-related obesity and its associated risks. Recent research from City of Hope has uncovered a novel mechanism driving the increase in belly fat during middle age, revealing a new stem cell type, known as CPAS, that plays a key role in this process. This discovery opens up new possibilities for therapeutic interventions, but it also raises important questions about how to effectively address the negative consequences of these cellular changes. The study, which sheds light on the cellular processes responsible for age-related abdominal fat accumulation, highlights the significant role of a type of stem cell called CPAs that emerges during middle age. These stem cells have the ability to convert into fat cells, contributing to the expansion of abdominal fat. This process, driven by a signaling pathway known as the leukemia inhibitory factor receptor, LIFR, leads to an increase in the production of fat cells, particularly around the belly. Although this discovery provides valuable insight into the mechanisms behind age-related weight gain, it also underscores the complex and challenging nature of combating obesity and its related diseases as we age. It is well known that fat cells tend to grow larger as we get older, but what's more troubling is that the body appears to have an unlimited potential to generate new fat cells, particularly in white adipose tissue, WAT, the fat responsible for weight gain. The City of Hope research team, led by Dr. Cheong, Annabelle, Wong, discovered that aging triggers the activation of adipocyte progenitor cells, APCs, which are stem cells that evolve into fat cells. In middle age, these APCs become much more active, leading to an increase in fat cell production, particularly in the belly. The researchers found that when they transplanted APCs from older mice into younger ones, the older stem cells produced a significant amount of new fat cells. This suggests that the aging process itself activates these stem cells, enabling them to churn out more fat cells, leading to increased belly fat. While these findings provide critical insight into the aging process and its impact on metabolism, they also bring attention to the challenges posed by the body's ability to generate fat cells as it ages. The creation of these new fat cells may contribute to the health risks associated with aging, such as diabetes and heart disease. The signaling pathway responsible for triggering the activation of these stem cells, known as LIFR, could be a potential target for future therapies. However, the fact that LIFR is involved in fat production means that any attempts to intervene could have unintended consequences, potentially disrupting the delicate balance of metabolic processes in the body. Blocking the formation of new fat cells may reduce abdominal fat, but it could also impair other essential functions in the body, raising concerns about the broader implications of such treatments. The discovery of CPAs and their role in fat cell production also underscores the complexity of addressing age-related obesity. While the findings offer hope for future therapeutic solutions, the reality is that the aging process is multifaceted and involves a range of factors beyond just stem cell activity. For instance, changes in hormones, metabolism, and lifestyle factors all contribute to the accumulation of belly fat as we age. Targeting a single cellular pathway, such as LIFR, may not be enough to effectively combat age-related fat gain and its associated risks. Furthermore, the research highlights the importance of early intervention. Preventing the activation of CPAs in middle age could be crucial in slowing down or preventing the progression of obesity-related diseases later in life. However, this raises questions about how best to intervene at the right time without disrupting other aspects of health and metabolism. Another key consideration is the potential for these discoveries to lead to personalized treatments for age-related obesity. While the research was conducted in mice, the findings have been validated in human tissue, suggesting that the same cellular mechanisms may be at play in humans. If future studies confirm these results, it could pave the way for therapies tailored to individual needs, targeting CPS and fat cell production based on factors such as age, genetics, and overall health. However, there are still many unknowns about how these therapies would work in practice and whether they could effectively prevent or reverse the harmful effects of belly fat without causing other health issues. In conclusion, the discovery of CPS and their role in the accumulation of belly fat during aging offers valuable insight into the complex mechanisms behind age-related obesity. While the research provides hope for future therapies that could help prevent or treat this condition, it also highlights the challenges of targeting specific cellular processes without unintended consequences. As scientists continue to explore the potential of sepia-targeted therapies, it will be important to consider the broader implications of these findings for human health. Although the discovery is promising, much more research is needed to fully understand the potential risks and benefits of these therapies and to develop effective strategies for combating age-related obesity and its associated diseases.